So, in this section, we're going to be talking about one of my favorite things, which is population and community ecology. And when you're studying different populations, and you have different reasons to do that, um, you can study it several different ways. So, ecology is the study of organisms with other organisms and usually with the physical environment that they're in. So the different types of ecology, just and these are just a few of them. You can do like spatial ecology, where you see how things are spread out. You can do organismal ecology, where you study an individual plant or an individual animal and how they react or behave, and their individual biology. Then there's population ecology, where you study a whole group of the same species, like several organisms of the same species, and then what they're doing and how they're interacting with each other. Or you can do community ecology, and community ecology is where you study several organisms and how they're interacting together, different species. You can also study ecosystem ecology, where you study all of the living and non-living things in an area and how they affect each other. So here's a picture, of those. To recap, you could study like individuals, you could study populations. That means the same type of species, but a bunch of them. You can multiple different species and can be completely different. Like um, that would be a community, or an ecosystem includes even the non-living things like soil or water or sunlight. And then you can study a whole biosphere, and this isn't even inclusive of everything. So when you're looking at them and studying them, it's important to know the population size and the population density. So population change size all the time, and it fluctuates. It can be due to human encroachment, or human disasters, or just natural disasters like earthquakes or floods. Sometimes you even have species compete for the same food source, and so that can cause fluctuations. So demographics describe populations and investigate how they are changing over time. And then life tables really try to show what the life expectancies are of individuals within populations. And both of those were really originally meant to target humans, so that they could track humans for various purposes. But then it's really been good to help with conservation, to know what's out there and to know the health of what we're conserving, and how different wildlife is being impacted by humans. So, a population size is the total number of individuals, and a population density is how many individuals are in a particular place. So, for example, desert plants are laid out almost like somebody planted them. I mean, it's like they're almost perfectly spaced. Um, it's just and really it's because, like, these plants, say these two plants here, if a plant tried to go in between them, they wouldn't have a big enough root system to gather water, and these would take any water that's there, because they'll have roots, you know 10 times or more larger than what you see above ground. They'll have underground, and so they have the root systems to go and get that water and take it before a new plant would ever even be able to come in. So these are very well distributed. Then you look at like rainforests, and they are totally clumped, because there's a lot of resources, so everything can grow on top of each other. So when you're counting and trying to do populations counts, there's a few different ways you can do that. The first way would be to literally just go in and count every single one. If in this picture, if you wanted to get every single one of this kind of plant, you can literally go in and count every single one in the space, and it's not a problem. They don't move, they're not going to run away. So you could count, and then you could count every single one of this plant there and get a census on that. But for things that move or things that fly, things that swim, that's really hard to do that. Or just even in very dense plant areas, it's hard. It's very hard. I've lived in these before and researched there. It's so thick and dense sometimes with vines that as soon as your partner walks away from you, like even a couple steps, they're gone. They've totally disappeared. You can't even hear their voice because there's so many plants that sound just gets lost. And so there's a few methods that you can use. Another one, besides just counting every single individual, is taking estimates. And one way you can take an estimate is by laying down what's called a quadrat. So that's where you lay down a square somehow. So a lot of times you do this in the rainforest, maybe with string, or you could lay down some meter sticks and make a board. But at any rate, you're laying down like a square area, okay? And you lay down that square, and then you count every single organism within that square. And then you take that square and you multiply it out by your space, and you can get a good enough estimate of what's in there. The problem with it is you obviously miss some, like for instance, this is one of the old growth trees probably in this rainforest, and there's not much old growth left because a lot has been taken away. So like if you laid down the quadrat right here, you would be missing the two old growth trees that are still left in the area. So it's not totally accurate, though it still gives you a good starting point or idea. For things that move, you can, so this is a picture of me doing these types of data taking on whale sharks. And so I'll put these two pictures here side by side so that you can see these. So this is me, and you can see that the tool that I'm using in my hand is actually a camera. Okay, so when I do this, there's a process called mark and recapture. So mark and recapture is where you use a mathematical equation that looks like this. It's pretty simple. It looks much harder than it is, and it's not. Once you actually do the math, it's not a big deal. You catch a certain number of the individuals and release them, but you mark them before you release them. So you catch a certain number and you mark them in some way, like put a band around their foot or put a tag on the back of their fin, and then you release them. Then you wait a day or two, go back out, and then you catch a whole second group and find and look at how many of the same ones you recaught, and tag all the ones that you didn't get the day before. So you tag all of them, and then you end up dividing by the number that were marked on your second catch. So here, if the first day you said, we're just going to do 10 a day, you caught 10 the first day and you tagged them all. And then you caught 10 the second day, then you multiply 10 times 10 on the top. That's easy, 100, right? And then you divide it by how many on the second day already had tags and you caught them a second time. So say the number is out of the 10, say 2 had tags. So then you would do 10 times 10 is 100 divided by 2. You can estimate the population size of being about 50 in that case. So say you caught 10 the first day, 10 the second day, but you actually had like 5 that were already tagged that you caught a second time, then it would be 10 times 10 divided by 2. That's only 20. So now we took that number from being, we estimate there's about 50 in the area, to we estimate the number is about 20 in the area. So that's how that works, and it's an estimate, and the more you do the mark and recapture and the more often you do it, the better and more realistic count that you'll have. However, going back to these two pictures, I want to point out that a lot of times mark and recapture isn't even done with tags anymore, because say there's one organization that's tagging this whale shark, and then another organization wants to go tag this whale shark, and say another organization wants to go tag this whale shark, and say one of the organizations, has their data public so that any people that are looking to whale or catch big fish can actually tap into your conservation website and find out exactly where the whale is to go get it. So it's not necessarily the safest for the animal. So that conservation people are definitely working to make this as easy on the animal as possible. So you can see that my mark and recapture is just a camera. And then what we do with the whale sharks is here on the edge of the boat, you stand there and you will swim beside the whale shark. Somebody from the boat will take a photo of you next to the shark, and they know your length, and then they estimate the shark's length. And then also with my camera, I will take a picture of behind the first dorsal fin, because each of their patterns is totally unique, like a thumbprint, and take a photo of that, and then dive down underneath, take a photo of their genitals to know if they're male or female, and then you will use your GPS coordinates, which now is pretty readily available on all devices and phones, and then you take down the date, time, location, if it's a male or female, and you have its thumbprint. You upload that all to a password protected system like this system I can't even get into anymore because I'm not active on a whale shark project, but you upload all of that information, and then you know exactly where that individual was, and then different people that have access to that system can track that same whale around the world, and that is how the mark and recapture is happening. Interesting too, as researchers, we wait usually 2 weeks to upload our data so that that whale shark is long gone. So if somebody ever did hack into the system, they're not able, that animal wouldn't even be in the area anymore. At any rate, that's kind of how this is happening more often now. In some places, it is. This is a picture of a python doing mark and recapture in Borneo, and us heading out to do mark and recapture on pythons, and also on something called a slow loris. So this is me trying to find this slow loris, and you can see how hard it is. I did have a stressed out look on my face, because the slow loris that we found just before then was unfortunately digested in a pile of python poop. So it was a stressful day. We did find, in case you don't know what a slow loris is, we were trying to find these guys that hide and sleep all day and only come down once, at the beginning of the night and the end of the night to go to the bathroom out of the tree. They're very hard to find. So researchers do actually put on a radio tracker on these guys to find them. And what's interesting is we're finding that as we shrink the rainforest, I'll go back to the slow loris because they're way cuter than me trying to find them. As we shrink the rainforest, we're pushing all the leftover mammals to a smaller and smaller area, closer and closer together. And what's interesting is we found that pythons, for example, or crocodiles, different reptiles, monitored lizards, they're actually doing really well with that, because what we did was we concentrated their food source. So they're like thriving at right now as we're shrinking the rainforest, because their food source is just like concentrated for them. So they're, an animal that has been here millions of years before mammals, and we're seeing that as we destroy, habitat, that they're going to last a lot longer than mammals as a whole too. So it's just an interesting situation. So species distribution patterns can be, I'm going to go back to your pictures. Species can be random, where they're just kind of all over the place. It doesn't really matter. It's like wherever it went, it's fine. You could have areas with a ton of dandelions and some where it's more sparse. This is where resources are fine, and usually the seed is just like wind blown or traveled by water, and it just goes wherever. Then there's like herd animals or even plants that just drop their babies, like, I'm thinking of like an apple tree or something just right below it, and it ends up making a grove. So that's called a clumped distribution. And then the last one is uniform distribution, and this is penguins that try to stay like an even distance away from each other as they live and take care of their egg. And they're a lot like the desert species, where it is a very even distribution, a uniform distribution of plants. The last thing that we have a couple more things actually to go over, is survivorship curves. So a survivorship curve, well, I want to go over demography first. I skipped this, and I feel like we already talked about it a little bit, but I'll talk about it again. Um, a mortality rate is to find out how many individuals are dying of a certain species. So you calculate this at any given time. You calculate the number of individuals that are dying of a certain species and in a certain area, and then the number of individuals that are surviving, that are still alive, and you multiply that times 1,000, and you get the mortality rate. And so here is what's called a life table, and this will have like the age, the number that's dying, the numbers that are surviving, and the rate. And this is the life expectancy, or the average lifetime of those getting to this point. So the average right here from 0 to 6 months old, and this one I believe is about sheep. I believe it's about sheep. Yeah, Doll Mountain sheep. Okay, so on this table, individuals that were 0 to 6 months old, if they survived, then the ones that survived the first 6 months of their life had an average lifespan of about 7 years. So it's just interesting as you look at that. So survivorship curves, this is pretty important to figure out and understand this. So there's some animals or plants that will create thousands, millions of different eggs or seeds, but even though they're creating millions of them, the number that actually survive as time goes on is like really low. So it's almost like I'm gonna put out, 1,000 babies and just hope one or two survive. And the types of, different plants do this all the time, right? They make way more seeds than what they are. But for animals, this would be like fleas. Fleas can lay something like thousands of eggs, over their lifetime. Or fish. Fish will do the same things. Spiders will, lay a way more babies than most of us are happy with. And then there's other animals. This is really interesting, that it's kind of, well, let me talk about this one first. This one is a survivorship curve. We're going to look at the blue line right here that they have humans on. This survivorship curve will have animals, or sometimes plants, but usually animals, that will have very few young, but very few die until they reach a certain age, and then most of them just die very quickly. Where most individuals reach a certain age before they start to die. So this could be, a lowland gorilla. This could be an orangutan. This could be an elephant. This could be a giraffe. This could be an orca, where they just live a certain amount of time before they die, kind of old age. Okay. Both of those, I think, are are easy to understand. What's harder to understand is this line. This line is where the individuals have kind of an average-ish number, like maybe this bird comes and lays 3 eggs a year, and out of the 3, maybe 2 survive, but whether they make it through the winter or not, it's kind of just as steady, like if you live or if you die is equally lucky. So it's not like, oh, as long as I make it to 6 months old, I'm probably going to live till I'm 70. No, this is kind of like at any point you have equal chance of living or dying. So not as many species follow this survivorship line on the curve scale. So some species that do are often birds, like I said, and it's the pictures here with like nests and being pretty clockwork of laying a certain number of eggs. But also you can have, for instance, a great white shark. A great white shark is like this, where they'll pretty like clockwork lay a certain number, give birth to a certain number of babies every year, and they actually have like a live birth of the eggs inside of them, and then they come out. But they will actually, equal chance of living it or dying at any particular moment. So that's the survivorship curves.